This is a production of Cornell University.
Thank you, Andrew, for that wonderful introduction. We'll get to the arm wrestling thing here in a second. Thank you to the organizers of the seminar series for allowing me to come and give this talk today. And thank you for all of you coming out and giving me your time to come listen to what we've been working on for the last 8 years on RNA modifications. And I'll start at the very beginning of my lab to give you some perspective and start there. My one lesson to you based on my introduction from Andrew is do not arm wrestle ever. It is extremely bad. And this would be why I'm retired. It's because I have a metal plate running from the top all the way down to the elbow of my humerus. And I cracked that humerus arm wrestling on Christmas Day with my wife's family. And I never get to live that story down because we spend every Christmas with my wife's family. And they remind me of it every year when it happened 5 years ago. And I also have that nifty scar. Anyway, so from there, now you know my story. Don't arm wrestle. I'll try and tell undergraduates a few other stories as we go along here. But what I am going to talk to you about, I don't know why this is doing this. If this is driving people nuts, it might drive me nuts. We'll see. Let's try to get through it, okay? But what I'm going to talk to you guys about is RNA modifications, how they affect post-transcriptional regulation. Oh, thank you. It's because my hands. Okay, anyway. How they affect post-transcriptional regulation to affect various stress responses. And that's where I'm going to spend most of my time in today's talk. But before we get there, I just want to remind you what post-transcriptional regulation means. And it means this very busy slide right here, which is all of the processes that an RNA has to go through after transcription makes it, or actually while transcription is making it, including capping, splicing, polyadenylation, modification, which is where I'm going to spend most of my time talking about today, folding, because we have spent some work on this as well, and then how all of these new features of the RNA molecule affect this very, very important decision here, which is whether to translate and whether to degrade and when we should degrade. And we spend a lot of time thinking about this transition and how modifications affect that. And that's really where I'm going to be spending most of my time talking about today. I do have to say that when I started my lab a number of years ago, we were interested really in looking at 3 features of the RNA molecule itself. What the RNA looks like, how it folds, secondary structure, which is what you can see here. So this RNA region knows how to fold based on intramolecular base pairing interactions between this region and this region, leaving this region single-stranded. And we developed protocols to look at this, and then went through and thought, okay, well, we're very interested in looking at what an RNA looks like, but in addition to how it folds, it also has a cadre of proteins that interact with it throughout its life cycle, from that transcription through capping, through folding, and then through this ultimate decision here. And these proteins are known as RNA binding proteins. And so we sat down and we spent a bunch of time thinking, could we develop an approach to look at secondary structure and protein binding all at the same time? And the answer to that question was yes, because otherwise I wouldn't have said that. And we developed this approach here that we call PIP-seq, whereby you can treat with structure-specific RNA ribonucleases with those proteins still there, or after light denaturing degradation of those proteins. And so we can use these libraries to look at secondary structure, and we can look at these libraries to take an RNA-centric view of every region of an RNA that's bound by a protein throughout its life cycle, based on what proteins are bound with the RNAs and the various kind of conglomeration of the collection of RNAs and a collection of tissue. We can get a snapshot of kind of all of its RNA binding domains. And we've used that approach to look at secondary structure in the Abiotic nucleus, and also to go through and find new regulators of root hair cell development and a number of other different questions. And I'm going to tell you, if I get to it at the end here, we're starting to look at nanopore to look at modification in newly transcribing mRNAs. And we're also going to try and get back into the structure game to also look at co-transcriptional folding and how modifications affect that. But I might not get to that part of my talk today. What I am going to spend most of my talk about focusing on today is that third feature of the RNA molecules themselves. So I've talked about folding, not going to talk about it anymore. Well, I'm going to briefly talk about it here in a second. We've talked about RNA binding proteins, but RNAs can also increase their coding capacity through chemical moieties that can be added to the ends, including the canonical cap, which is an M7G, as well as polyadenylation, and how long these tails are can kind of dictate how stable an RNA is. And there's lots of internal modifications that can occur in specific regions, either the CDS, the 5' UTR, the 3' UTR. And what I'm going to be focusing on today is this modification itself, M6A. And I'm also going to be talking about this non-canonical cap molecule here, this NAD cap. And in fact, I'm going to start at the 5' and work to the 3' when we start talking about RNA modifications. We'll get to that here in a second. But I also just need to show you that I'm going to talk mostly about 2 RNA modifications, but there's about 153 modifications that we know of that occur on RNAs. We know this mostly because they occur on tRNAs and RNAs, which are easy to extract, easy to mass spec, and easy to get at. But what we are finding is many of these also do dot mRNAs. And so figuring out their functionality in mRNAs, obviously for you graduate students, there's still a lot of work to be done in this field. And in fact, you might even be able to select your own modification and go work on that in the future. So keep that in mind, right? There's still a lot of room in this area of research for many types of researchers to keep working on. But like I said, what I'm going to focus on quickly is our recent work on this non-canonical cap, this NAD cap that we find can replace the M7G cap many times, and it can specifically, on specific cohorts of transcripts, replace this cap to affect specific physiological processes. And I'll show you our evidence for me being able to say that here in a second. So please remember, if you remember nothing else from my talk today, please remember this slide, and that this is what a mature mRNA looks like. It has a 5' cap, a 5' UTR, a coding region, which most of us think about because this is where the protein is encoded, a 3' UTR, and a poly A tail. What I can tell you is this is genomically encoded, this is genomically encoded, this is genomically encoded, and both of these are added during transcription and post-transcriptionally. And in fact, it's the addition of the poly A tail that terminates transcription in eukaryotes, because it's the cleavage factor that sets the end of the 3' UTR to start polyadenylation that basically terminates transcription of that transcript in eukaryotic organisms. And so if you just want to know what the chemical structure actually looks like here, here's your stick and ball model here. And you can see that this is actually attached, this canonical cap, the 7-methylguanosine is attached to this phosphate-phosphate bridge, whereby it's the alpha phosphate from the 7-methylguanosine that actually attaches to the beta phosphate at the 5' end of the mRNA. And so you still have a triphosphate, but it's protected even more by the 7-methylguanosine. And this methylation event is actually put on later, and most likely is involved in self versus non-self. And we are getting quite interested in this type of methylation event here too, but I'm not going to be able to talk about that today. But we've also found, we and others, we weren't the first to discover that this modification could be added to mRNAs. In fact, bacterial scientists, or in fact, people working on bacteria as their model organism, found in fact that this NAD RNA, the nicotinamide adenine dinucleotide molecule, and that's the last time I'm going to say that because I said it perfectly and I feel very good about myself, because usually I say it completely wrong. And I'm just feeling very good. So we're just going to call it NAD from now on, okay? This NAD molecule can now be added, we found, to the 5' end of mRNAs. In bacteria, we know how it's added. So, because I know I'm going to get this question, so I'm just going to answer it now. No, we do not know when or how this molecule is added. In fact, this is something that we are actively looking at, right? Because we want to be able to control the addition of this, because we find that it's an incredibly powerful post-transcriptional regulator, and I'll show you evidence for that here in a second. But we do not know how this is added in eukaryotes. Okay, there you go. So we don't. So I would venture to say yes. This is something we're very, very interested in. Like, how does the overall metabolic pool affect what modifications can be added? Right, methylation events. If you are depleted for it, you're not going to methylate your RNAs anymore. What I can tell you, because I cut this part out of this talk, is we found if you trap this on the end of an RNA molecule, the plants get very, 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 very unhappy. Okay, so if we inhibit the ability of the plant to get rid of this from the end of its mRNAs using the enzyme that normally does it, which I'm going to introduce to you here in a second, DXO1. So if we knock that out and keep it on there, plants will actually go around that and start chopping those mRNAs into small RNAs, 21 to 24 nucleotides in size. So what it looks like, in fact, is for all of you small RNA biogenesis aficionados and people that are interested in that, it looks like small RNA biogenesis actually was an ancient degradation pathway that has been co-opted now to make small functional RNAs. But its first probably introduction into eukaryotes was just to chop up RNAs. And that was the first RNases that they had. And it broke it up into bigger pieces and single nucleotides. So then what happens is if you keep this on there by knocking out DXO1, the denatter, plants will go through because they really want that NAD back to get back in the pool of metabolites. They'll just break it down into small RNAs, thereby recycling through additional means. And if you knock out the ability to make small RNAs, then they actually upregulate all the enzymes to make NAD. So I'm assuming that if you have NAD deficiency, something's going to go and you're not going to properly regulate your transcriptome. So I'm going to show you here in a second. Plants use this to specifically destabilize pools of RNAs, depending on where they are in development and where they are in responding to specific hormones. And I'm going to come to that in a second. Did I answer your question well enough? Yeah. Okay, great. Very well. Okay, so our first foray into this, and of course, I can tell you this in a very beautiful linear talk, and you're like, oh gosh, yes, you were so brilliant. You thought of this, and you guys just found the mutant and then just went through and characterized it as poppycock. This next little vignette I'm going to talk to you guys about, I started as a brand new professor when I read the first paper on this denatting enzyme. It was called something else in yeast, but I went and found the ortholog in plants. And I worked on this myself 8 years ago. And it took us somebody else publishing that this is, in fact, a denatting enzyme. And then we went in and said, okay, well, let's make an approach where we can look at NAD-capped RNAs in plants. And then finally, we could put 1 and 2 and 2 together to figure out what the heck this enzyme does in plants and what this moiety actually is doing within transcriptomes. And so then we could add to this body of literature, mainly because we had all of this stuff laying around because we've been working on this project for 10 years. So graduate students, don't give up, right? Never give up. Always remember your data and just keep plugging away because eventually something's going to help you, something's going to click, and you're going to end up getting a very nice story, a very nice publication out, even when the days look bad. So what I'm just telling you here is we developed an approach to specifically pull down the NADylated, the mRNAs with NADs on their 5' ends, and then we could clone and sequence those using Illumina sequencing. And so what I'm using this slide here is to show you, in fact, that the NADing of RNAs is very, very specific between tissues. So if you look at the NADed RNAs in 10-day-old seedlings and compare those to the ones that we find in wild-type plants here, so these have all the functional components of NAD, adding the NAD and denatting the transcripts, you can see that it's basically an entirely distinct pool between these two tissues. And what I'm going to show you, even if you treat with ABA for a few hours, we can find very distinct pools. And that's why we think the addition of NAD in eukaryotes is not done transcriptionally, just by set down by that first nucleotide, but is in fact an active process whereby you can actually potentially remove the NAD and recap, or at least it's added as same as the cap in eukaryotes, where it's an enzyme that adds it to the 5' end during the process of transcripting. And so what you can see down here, and this was the enzyme that I was getting at, DXO1, this is a denatting enzyme conserved from yeast to humans to plants. And when we knock it out, we get the expected phenotype when it comes to NAD RNAs. Specifically, now we can find more RNAs in their NADed form because it's stabilized in that form. Like I told you, the plants don't like this mutation. They go through and they start turning these mRNAs into small RNAs to get that NAD back. So this is a very sick plant, but we use it as a tool to find those NADed RNAs and to specifically stabilize them, and also to go and do some interesting biology. And so then what we also found, I cut the slide out here due to time constraints, but what we did was when we did these original flower bud sequencing, we found that ABA was one of the most overrepresented terms in normal wild type tissues for those RNAs that were NADed. And so that made us think, oh, well, that's pretty interesting. So what happens then to the pool of NADed RNAs in both wild type and DXO1 mutant plants if now you look at NAD before and after ABA treatment? And this is 4 hours of ABA treatment, and then we take the RNAs pools and then we do our pull down and sequencing. And so what you can see is obviously there is a collection of RNAs that are NADed both before and after, but we can find a huge collection, or at least 600, almost 600 transcripts in wild type and over 1,000, almost 1,100 in the absence of DXO1 function that are NADed only before treatment. These then don't show that same NADylation or the NAD on the 5' end after treatment. And now we find a whole new collection of molecules that are actually natted specifically during ABA treatment that were not natted before ABA treatment. And so what I'm going to show you is the first thing that we decided to do is figure out, is adding a NAD stabilizing, destabilizing, or are RNAs agnostic to having that molecule on them? And so what I'm going to tell you is this was an approach I developed as a postdoc. I never knew how wonderfully useful it was going to be. I literally just did it to say that if you didn't have a 5' cap in plant transcriptomes, you're going to be turned into small RNAs. But what we found is this is what we call GMUC, or genome-wide mapping of uncapped and cleaved transcripts. I didn't know it at the time, but Mike Axtell developed a similar approach. He called it degradome sequencing. Blake Myers and Pam Green developed a similar approach. They called it pair. My name was way too many words and way too complicated. And so pair is what you guys call it today, basically, or what most people call it, either that or degradome seq. But GMUC is actually basically the same thing as degradome seq or pair. The one thing that's different is they actually put in their initial library prep, they put a restriction enzyme binding site into their 5' adapter. And so their reads are all very, very short inserts. We figured out a way now more recently whereby you just put on the 5' adapter. So it's 5' race. It's genome-wide 5' race where you put on a 5' adapter, and then you select 3' ends randomly by basically just using an RT primer. That's a random hexamer attached to the 3' Illumina adapter. And we can now make these libraries in a day's time from beginning to end, and we have 5' race. And so what I can tell you is you can use this approach to look at endonucleolytic cleavage. I'll show you some evidence to show that M6A actually inhibits endonucleolytic cleavage in Arabidopsis. I'm going to show you how we use it as a quick and dirty mechanism to look at, are you more degraded or less degraded, like with ABA or without ABA, or with NAD or without NAD. And I'll show you how we use it that way. And we also found that there's a co-translational decay pathway that functions in plants. And so we can actually use this approach now to also look at translational dynamics in plants, and also to look at the transcripts specifically that are degraded co-translationally. I don't think I'm going to have time to show you any of that data, but I'm happy to talk to anybody about that if they're interested in it. Okay, so what we actually found is this proportion uncapped metric. It's just a quick and dirty measure such that we don't have to treat with transcription inhibitors every time. We can actually just correct GMUC read total libraries for a transcript over the amount of reads that we see from mRNA-seq. As long as you take the same sample, do GMUC on half, do RNA-seq on the other half, and then you can use this just correction to give if we have higher proportion uncapped metric, that means in that condition, those RNAs were more degraded. If we lower that metric, then in those conditions, those RNAs were less degraded, okay? And so what I'm gonna show you is, in fact, we use this metric for NAD-capped RNAs that were only capped in control, capped in both conditions, or capped only in ABA conditions to show, in fact, and we did this multiple times in multiple different conditions to show that that NAD molecule being added to the 5' end of transcripts is an incredibly destabilizing moiety. And this is exactly what this type of data then looked like. Like I said, we had those transcripts that were only NADed in control in both conditions, and only those in ABA. And so what you would expect is, if it's a destabilizing mark, because what we're doing here is we're correcting that proportion uncapped metric in the ABA samples. So what we should see is that metric rise when you add it in the ABA samples. When you're comparing ABA to control, we should actually see that metric decrease in the ones that are only NADed in control, because now in the ABA conditions, which are in the numerator, we should have no NAD being left and only the NAD being left in the control. So the higher proportion uncapped would be here versus here. In this case, the higher proportion uncapped is here versus here. So we see an increase, and those shared were centered right on 0, whether it was in wild type conditions or those DXO1-2 mutant conditions. And so what this told us, in fact, was yes, this moiety is, in fact, an incredibly destabilizing moiety when it's added to the 5' end. And so when you do have it, you're going to destabilize those RNAs. When you remove it, now you're going to stabilize them. So this pool becomes stabilized in the context of ABA. So then we were interested to just kind of say, what type of what type of transcripts do we actually see with that NAD molecule in the control conditions versus after ABA treatment? And this is what we were hoping to see. And thankfully, this is exactly what we saw. So what these plants are doing is actually using that NAD molecule on the 5' end to destabilize those transcripts when you don't want them around. Then they remove it either through transcription or removing it and then recapping, which does happen in plants in the cytoplasm, I might add. And then in this condition, now we found that these regulators were now not natted and were actually stabilized such that you could make the protein and get proper ABA response. And so I can tell you this is exactly what it was. We went and looked at some of these important regulators. Don't worry about what's going on in DXO1. DXO1 is incredibly ABA insensitive. And this is exactly why, is because those regulators are not properly regulated. But for instance, one of the master regulators of the ABA response, ABI5, is natted in control conditions and no longer natted when you treat with that ABA response. And what I can show you then is when you can go through and specifically validate this, doing the NAD versus the NAD versus the non-NAD fraction in control versus ABA in both Columbia and DXO1. And we can show that when the cap is on there, they're destabilized. When you remove that cap, they actually now become stabilized, they stay around, and they actually make the protein to give you proper ABA response. Think of this model right here, whereby, in those control conditions, right, you have the NAD on there, you denad, and in fact, this enzyme most likely is involved in the decay as well. So you keep those RNAs at low levels such that you don't have ABA response when you don't want it. When you add that hormone ABA and you want to have proper ABA response, you actually add the right cap, either through transcription of new stuff and proper capping, or by removing the NAD cap and recapping of the cytoplasm. That's what we're working on right now. This then inhibits the denadding and the decay. And so now you make protein, you get proper ABA response. So this is what we're working on in the context of NAD capping. And like I said, we're very interested in how it's added, how it's controlled, and also looking at other things involved in this denadding process in the context of ABA response right now. So I've talked to you about a 5' modification and how this is a destabilizing moiety. The modification that actually got us thinking about this feature in RNA molecules themselves was actually this one down here, this specific internal modification, which to date is the most abundant internal mRNA modification that we know about, specifically M6A. And I will say, once again, mammalian systems were characterizing this over and over and over again, but there actually hadn't been a very good study about what the function of this modification was in plants when we first started down this road. So we collaborated with Rupert Frey, and before I get there, I just want to remind you that because work has been done on this system, there's even more known about it, right? We know that this is exactly what adenine looks like. We know the entire complex of proteins now that's involved in writing. There's actually 2 of them. The one I'm going to focus on today is the major one that's driven by MTA, which is the ortholog of metal 3 in humans, which is the major methyltransferase in humans. So we know what writes it or adds it to the adenine molecule. We know what erases it. And I'm going to talk a little bit about these erasers down here, because if you think about it, there's 2 ways to make a hypomethylation type of state within a plant transcriptome. You can either knock out a writer, which would then get rid of this modification, or you can overexpress an eraser. And thereby that also would remove more M6A than you might want to. And so I'm bringing this up mainly because I'm going to show you knocking out writers many times. But in our most recent study where we were looking at pathogen response, we actually used both of these strategies, knocking out parts of the writer complex, as well as overexpressing erasers to get at what truly hypomethylation looks like in the context of pathogen defense response. Don't worry, I'm going to show you far less data moving forward and get just more to models, because a lot of this has been published for a long time. That's why I can move through a lot of projects moving forward here now very, very quickly. And I will tell you, we're also working on some of the readers. The readers that we're most interested in are those that are stress responsive, specifically show increase in abundance during specific stress responses. And the ones that we're working on are the ones that are upregulated in the context of bacterial or biotic stress response, specifically during bacterial infection. And I have a graduate student postdoc working on some of those, but I'm not going to talk about that today. So when we first got into this game, we have to get past the fact that M6A, or methylation of adenine at the 6th position, is a fundamentally important modification in plants and animals. If you knock out those writer proteins, you are embryonic lethal. So the plants are dead at their embryonic stage and you can't really characterize them. So thankfully, Rupert had been working on this for a few years before we got interested in this modification. And so he had developed this nifty little plant line down here where your homozygous knockout for MTA, but we can get past that embryonic lethality by expressing MTA under an embryonic specific promoter, thereby getting us past that block. And then we can get into development. And depending on the levels of M6A that you allow left, these plants don't look normal. This is probably the best picture we ever took at one of these things, but you can already see they're smaller, they're purple. And I can tell you really when you grow them on soil, they look very, very sick. So this is a very nice system, and we can get the levels very, very low. And specifically, we started looking at leaf tissue because we could keep the levels of M6A lowest in that specific tissue. So our first question was, is this a stabilizing or destabilizing moiety? So once again, this is where we first started using that trick of using GMOX, our 5' race protocol with the correction to show, right, that we can use this as a quick and dirty approach to look at RNA stability. We also then developed M6A pull down. We weren't the first one to do it. We just modified it so that it worked well in plant transcriptome. Specifically, we're using M6A specific antibody to pull down those methylated regions and do high throughput sequencing in plant transcriptomes to show that. And then we and others had showed, right, that this occurs mostly in the 3' UTR and around stop codons. And if you want to see what the patterns of methylation look like, you can read any other paper. But what we were interested in saying is it stabilizing or destabilizing? So once again, we used wild type versus MTA plants because basically based on what happened in these, if we see an M6A deficient types of transcripts, do they become more or less abundant in the case versus wild type? If you look specifically with transcripts that have M6A sites versus all other transcripts that aren't M6A, at least in our data site here, what we found is in fact, when you knock out the major methyltransferase, these transcripts then become destabilized. Specifically, those methylated transcripts become destabilized in the absence of methylation as compared to when the methylation was present. So unlike NAD, now what we were seeing was that M6A was a stabilizing moiety. And so I'm happy to report that we weren't wrong. And many other groups have gone on and seen the same thing in the context of many other different plant responses. And in fact, the human people realized that they weren't analyzing their data wrong. They were analyzing their data wrong. And in fact, M6A, unlike earlier reports, is a stabilizing moiety also in animals. And why is it a stabilizing moiety? Well, what we found is when you go back to that GMOX data, like I said, we can also look at endonucleolytic cleavage. What in fact we found was if you look in M6A and you look 4 to 5 nucleotides upstream of the site within the motif that was methylated in our peaks, we can in fact see that this methylation event tended to be 4 to 5 nucleotides downstream of a very strong cleavage site that we can map by looking at the very 5' end of our GMUC reads. That's the most informative nucleotide in all of GMUC because it tells you right where the cleavage event occurred. And we could say, we see a cleavage event in this thing, and now we see that the methylation site is 4 to 5 nucleotides downstream. And when it's gone in those M6A deficient mutants, specifically in MTA mutants, now what happens is that methylation event is gone because they're M6A deficient. You get an endonucleolytic cleavage. We now know what this is in humans, and most likely the same ortholog in plants does this types of cleavage event. And then you get clearing of these intermediates by XRN4 and most likely the exosome once this cleavage occurs because you've lost that methylation event. Okay, so this happens both in plants and humans. How M6A is stabilizing is basically when it's there is inhibiting an endonucleated cleavage event, 4 to 5 nucleotides upstream of that methylated A. And so when you get rid of the M6A, now you get the cleavage, and then you get turnover of those RNAs. So I can tell you in the context of this study, what we also started to look at was, is this type of methylation event, does it have physiological relevance, okay? And so what we first started working on, and I'm going to show you now 3 different abiotic stresses. We first started looking at abiotic stress to say, is M6A important? Do we need it there to get proper response to salt, cold, and heat? More recently, heat. I'm going to show you no data for heat. I'm going to show you some data for salt. I'm going to show you some data for cold, and then I'm going to show you some data for biotic stress as well. And then I'll be done with M6A as well. So what we've actually turned to looking at was when we looked at methylated salt stress response encoding protein mRNAs, what we found, in fact, is when you had methylation events in their 3' UTR, we also saw changes in secondary structure. So in fact, the methylation event was there to decrease secondary structure. And specifically, it was only these specific salt stress responsive transcripts that showed this change, both in having methylation now in salt stress and the structural change, and not any other type of transcripts then that led to increased, really screwing up with this pointer here. It was only these transcripts specifically that were methylated with a structural change that showed increased protein output in the context of salt stress response. And so it led to this specific model here, whereby that methylation event had to be there to most likely unravel a structure, because it's a structure busting type of moiety. If that structure was forming, this was most likely what was recruiting that endonucleolytic enzyme, giving you cleavage, leading to turnover and destabilization. Then if you wanted to have proper salt stress response, what happened is you methylated these transcripts encoding the proteins involved in salt stress response. We inhibit then the recruitment of this cleavage enzyme, stabilizing these mRNAs. And it's only these ones that also change the secondary structure that led to increased protein output from these salt stress response regulators, giving us proper salt stress response. So because I talked about structure, I'm just going to tell you one other little vignette, 2 slides, I promise you here, mainly because we have used structure data in the context of hypomethylation, once again, in those MTA mutants, as compared it in normal methylation, to show that you have to have proper methylation of primary microRNA transcripts. If you don't have proper methylation of these transcripts, they fold into weirdo types of secondary structures that we could make models for using our data-based approach. And thereby, if you don't make the proper stem loop, you obviously don't cleave out the microRNA. And so then you also don't have proper microRNA biogenesis. So then that's what you can see here. Basically, you need proper methylation to get the proper stem loop folding. And if you don't have the proper stem loop, then you don't recruit the biogenesis machinery, and you are deficient in specific classes of microRNAs based on this type of structure and methylation of the data that we've made. Okay, so as I told you, we had done one abiotic stress, specifically salt. So we next turned our attention to whether other abiotic stresses could also be involved, or whether M6A could be involved in other abiotic stresses. Specifically now, we treated with cold for 24 hours. Once again, what we did here is we looked for cold-specific peaks versus peaks that we only saw in control conditions, and we did shared peaks. And what we found specifically is when you looked at transcripts that were methylated only in cold and showed an upregulation of their mRNA abundance, now that's where we found those cold response protein encoding transcripts that were specifically methylated and upregulated in response to cold, and they were found in this transcript here. This can be shown here, whereby when you look at cold enriched, this is just for mRNA abundance here. When you look at cold enriched peaks versus cold enriched peak, control enriched peaks, and shared, what we can see is that these become stabilized specifically, methylated and stabilized specifically in the context of cold response as compared to those controlled conditions. You can see once again, there's no movement and no shared because they are methylated both in the context of control and cold conditions. We also decided to say, because we didn't see any changes in RNA stability, I'm cutting out all of that data. So we did some ribosome profiling to say, is there an effect then on the level of translation from these specific cold stress response encoding proteins? Since I'm showing you the data, the answer to this question was obviously yes. And you can see that data over here. Specifically, those cold enriched and increased abundance transcripts were more ribosome enriched or more ribosome bound and gave us higher translational efficiency as compared to those that lost methylation or had methylation both before and after cold treatment. And so what this told us once again is in the context of cold, instead of inhibiting cleavage, now what you are doing is this methylation event was somehow increasing the protein or the translation of these mRNAs. And we're still looking at how methylation is doing this, because unlike in animal systems, which I don't believe that data as well, they said that if you methylate here at the 5' UTR, we saw no 5' UTR methylation. Okay, they said that the methylation event could actually recruit the translation machinery itself. We don't have any evidence for that right now. We actually think that what this methylation event is doing is it's still here in the 3' UTR, because that's where we see all these cold specific peaks. We think it's actually helping to just cause a circularization, and then the function, basically just keeping those ribosomes better equipped through the YTH proteins being bound to this specific methylation event, but I don't have time to show you evidence for that today. Okay, so I've shown you that we can see that M6A is important for salt stress response, cold stress response, and what I can tell you is we're about to publish on plant heat stress response, which looks very, very similar to the salt stress response, whereby it becomes a stabilizing moiety, thereby increasing half-life, which I'm going to show you is also the case in biotic stress response. So far, only cold is where we see this translation phenotype, and so this is a pretty cool stress that we're continuing on for that translation effect. So once again, I just want to remind you that you can make hypomethylation by knocking out writer components or by overexpressing eraser components. And I'm just telling you this mainly because we used both types of either mutants or overexpressors to show that it's not just the phenotype of the eraser mutants because they're small and they're weird. These plants look very, very normal when we just overexpress one of these erasers, even though they're hypomethylated, but we can still see that they have the exact same pathogen defense response defects, which I'll show you here in a second. Okay, so first of all, what we found, and this is how we got into this game, was when we just looked at bulk M6A and we treated with either bacteria or mimicked bacterial infection by treating with the FLAG22 peptide, which is the 22 amino acid peptide of flagellin, which is one of the potent signaling molecules that plants are looking at. What we found was that there was a large-scale rearrangement of the M6A in very, very quickly in response to either pathogen infection and or FLAG22 treatment, and then that was corrected by 24 hours of either infection or a little bit later on after flag treatment. So plants were basically wiping their M6A transcriptome and then putting it back on a little bit later. And so what we decided to do here, once again, in the context of two different ways, in this case, I'm just showing you the data for overexpressing one of those eraser proteins. We also looked at hypomethylation and MTA and found that many of these transcripts that were hypomethylated either in the MTA mutant and or in the overexpression of that eraser overlapped both in control and in FLAG22, basically mimicking bacterial response. And what in fact we found was the things that tended to be misregulated before or in control conditions as compared to after FLAG22 treatment, in either of these hypomethylated conditions had to do with response to bacteria, and response to fungus, and response to biotic pathogens. And so we were continuing down this way. What I'm going to tell you is kind of skip over a whole bunch of other experiments to tell you what we found was using GMUP once again. It was stabilizing defense response encoding protein, defense protein response encoding transcripts. And so we just validated some of these things using the common way of inhibiting transcription, which is why I like our GMUP approach better, because these type of experiments are incredibly labor intensive. You get data for one transcript at a time, but this is exactly the data here, whereby when you have a hypomethylation event, these regulators are actually then destabilized in the absence of methylation when they should be much more long-lived when you treat with FLAG22 for these specific defense response regulators that I'm showing you here. And so just to show you some physiological relevant data here, what you can see is both in the context of a methylation mutant. So this is the methyltransferase, we can make the mutant there. What you can see is they're actually resistant. They're more resistant to both pathogens, as well as they don't show this growth inhibition when treated with that FLAG22 peptide. And they are also more resistant to different bacteria, as well as if you treat with a fungal pathogen. So in this case, getting rid of the methyltransferases and or overexpressing this eraser protein and making a hypomethylation event makes you actually more resistant to a biotic, specifically a bacterial pathogen, and or treatment with this FLAG22 peptide. And so we're very, very interested in this result right here, especially, mainly because I didn't have time to show you this today, but these plants actually are slightly bigger than wild type. They develop slightly faster, and they are more resistant to bacterial and fungal pathogens. So we think, in fact, this type of hypomethylation event might actually be good from a crop improvement perspective. And so we're going to try and put this into other plant species and actually test this in a relevant crop species. And so what I hope I've convinced you of today is that M6A is an important post-transcriptional regulator of both plant abiotic and biotic stress responses. And we're continuing to work on this on the context of the eraser proteins in the context of biotic stress response as well. I have 5 more minutes, I believe, for this talk. And so I'm just going to tell you very, very quickly, because we're actually collaborating now with Andrew on this last little vignette that I'm going to show you here. And I just want to remind you, based on this slide, that I've talked to you about a lot of data for this type of 5' cap, non-canonical modification on the cap structure. I've talked to you about this one modification here, and you can see how important these 2 modifications are. But just to remind you, right, there is an incredible collection of modifications still out there to be characterized and looked at, most likely from a lot of different physiological types of perspectives. And pulling down one and sequencing one in a specific type of developmental and or pathogen and or stress context is incredibly labor intensive. And it's going to take a lot of work from a lot of different investigators to actually do this type of work. Plus, you have to use a very specific type of antibody or chemistry to specifically turn NAD into biotin. And there's a lot of work to look at one modification in one type of perspective. And so to be quite honest, this was our first project in the modifications. Then we got really interested in M6A. And then we got, like I said, we were working on NAD for 8 years and finally figured out that it was denadding that was important in the context of the DX01 mutants. But what we had also thought about was, can we come up with a much quicker way to find modification sites that does not require pull down, does not require any type of fancy antibody-based sequencing approach. And nicely for us, RT, specifically non-error correcting RTs, reverse transcriptases, do nature's work for us. Specifically, when they find modifications that affect the Watson-Crick base pairing edge, that is the chemical information they're using to put in the base at that specific nucleotide, such that then when they see one of these modifications that affects those 3 positions in GC base pairs and those 2 positions in AT, so it's about 45 modifications now, if they don't error correct, you can just go through any type of RNA-seq data set looking for these types of sites, specifically mismatches or sequencing errors at those sites, and you can find modification sites. And we call this algorithm HAMMER, or high throughput annotation of modified ribonucleotides. We've done it on small RNA-seq, we've done it on GMUT, we've done it on RNA-seq, and this is exactly what it looks like, whereby it's a G, and you can see a pileup specifically of misincorporated bases there, and we can call that site then an 2-methylguanosine, right, or an M2G site, and we can see about 45 of these specific, as long as they affect that Watson-Crick base pairing edge. And because these mutations are actually semi-random, M2G, you get a very specific collection of those base mismatches. Same thing for M1A. We can actually kind of subcategorize them into about 10 different groups, such that we can turn 45 modifications into 10 different subgroups, and do this just going back and re-annotating now all of the RNA-seq data sets from every different crop species. And this is exactly what we're working on with Andrew right now. I didn't have time to show you any of this data, but this is the question that we were also interested in asking with this project, specifically because when we did this approach in Arabidopsis, what we found, in fact, is that modifications tended to be found much, much more often in the GMUT library when we just looked at these types of modifications as compared to normal mRNA-seq libraries. And so what it looks like, in fact, is that the pool of degrading RNAs gets modified by this type of modification that affects this Watson-Crick base pairing edge as they're being degraded, such that they can never be translated ever again, and they just go into the degrading pool. And so we're trying to answer this from an evolutionary perspective with Andrew currently. So once again, using this algorithm now, just analyzing any RNA-seq data set, we can get about 45 modifications. Sorry, M6A doesn't work because it's at the 6th position. It's not one of those the 2 positions in A and T that affects that Watson-Crick base pairing edge, but at least we have an approach to get at that. And we can't look at in modifications, I should say. So once again, we're stuck with NAD. You have to pull that down and sequence just that. Okay, and with that, I just want to cite the people in my lab. These are the current members in my lab up here. Lisa and Will are two great graduate students. I have two great postdocs. Ron is still a graduate student, a great group of undergrads, and I just found out that I left Will's name off there. Will Prawl was the one who did all of the work on the biotic stress responses. I also cited down here all the previous lab members whose data I talked about from the published perspective. The Kresge lab and these peeps here were helped us with the abiotic stress response and are now working with Will on the demethylases. And all of my funding so far has come from NSF, both MCP and ILS, and Will was supported by a Fulbright scholarship. And with that, I'm going to stop. I'm happy to take your questions. Thank you for that. Yes.
Do you have any convinced about the RNA? And I want to ask you something again.
Yeah.
So you showed the picture of the side modification, and I'm interested in because of the COVID. This is side modification. Have you looked at it?
So that's pseudouridylation, right? And yes, so pseudouridylation, thankfully, we can see it by hammer. And it's definitely there in specific mRNAs. We're right now, because we found in a specific context, we can actually increase the amount of pseudouridylation we can see in specific mRNAs. I didn't have time to talk about it today. And so now we're actually going back and using some of our structure sequencing approaches, because it's always been said that that's a structure stabilizing moiety. And we want to see, is it structure stabilizing? And do you have to structure stabilize to actually see the importance of it? So yes, it's a great question. Yes, it's in plant transcriptomes. Yes, we think it's important, but I can't convince you of that yet today. So I just didn't put it in my talk. Margaret.
So in your DX01, you're seeing basically phased small RNA production as a degradation method.
Correct.
Do you see a role for NAD capping in functional phase RNA production? So
I have asked Blake to look, and I'm sure he's looking. He hasn't gotten back to me on that, but I mean, he's a phasey guru, so I did not want to touch anything to do with that. But I told him, you know, I showed him this data a while ago, so I think he's well on it. I was reviewing, I think one of Keith Slotkin's papers, and he was talking about this, and we had just published this. I'm like, you should speculate to remind Blake, and so he cited the paper and speculated, is this involved in phasing? So great question. Yeah, at the same one. I'm going to avoid Andrews because I saw your hand up first.
So a question about the NAD caps and ABA genes are overrepresented.
Yeah.
So my first part of the question is, are those ABA biosynthetic genes or are they response genes? And then I'm running the risk of overinterpreting, but in terms of the other modifications, M6A, like heat, salt, pathogens are all things, especially the pathogen lifestyle, be influenced by ABA. So am I overinterpreting, or is there some very specific effect of these modifications? RNAs involved.
So I can tell you we have not looked at ABA in the context of M6A. My guess is yes, there would be some effects on that as well, just because M6A marks all these things. And every time we look, you kind of wipe it off of the things, you put it onto the things you want to stabilize in that specific condition. And that's why it's a beautiful little mark, right? Is because you're adding it co-transcriptionally, we believe. I didn't have time to show you our data for that, but that's one of the major things we're working on, is we do believe it's added co-transcriptionally, and we believe it's the erasers that are selecting the sites. And so that's why Will's working on the erasers right now. Methylation is actually happening everywhere in a transcript. If you read our biotic stress response paper, you can see the hints that we were going on. Specifically, when you knock out the erasers, now we start seeing methylation all throughout the CDS. So we're now learning how to purify and sequence nascent transcripts so we can show that in the context of the eraser mutants that is happening co-transcriptionally. That's the last thing we're doing right now. But we think it will affect ABA. We don't know, we haven't checked for NAD capping in the context of other stress responses. That's actually part of a grant I'm writing right now, mainly because we do want to look, because it does look to be such a potent destabilizer. But please remember, what we think is going to happen then is NAD is going to have the opposite effect. So Growth regulators are the things that are now going to get natted, for instance, in the context of stress response, because we know those become destabilized, right? So we think that the growth regulators in the context of pathogen infection are going to get natted and destabilized, whereas the things encoding the pathogen response proteins, and I just showed you this, get methylated and now they're stabilized. So we think it's these two modifications are actually going to interplay off each other to give you that switch between growth and stress response. That's a great question. This is exactly where we're heading and thinking in that regard. Great question.
So I think you might have just answered something that was eating at me through the NAD part of your talk. Seems like preventing turnover sounds very waste, right? But it sounds like you were just saying that nadding is actually a regulatory in your mind, or maybe in your test tubes, is actually a regulatory mechanism. And so I should be thinking of it as turnover as being the thing that's regulated, rather than wasting all of this RNA and to nucleotides and oligonucleotides.
Correct.
And then turning that off.
Right. I think that's, think about it from the degradation perspective. Sorry. Don't think about it from wasting nucleotides perspective. Yeah. That's how I don't think about it from the wasting nucleotides perspective. Andrew, you want to ask?
It's not a plant question, but I was curious about the difference in response M6A and the abiotic stress versus M6A and biotic stress. And I was wondering, with the biotic stress conditions, that's where you have foreign RNA. Do you think like that is contributing to like the difference, or do you think you have another model?
No, I mean, so this is based on not work that we've done in my lab. It's the viral, right? It's the viral work from other labs, whereby viruses actually use the plant's M6A machinery against them, because they take the machinery, methylate their transcripts, stabilize their transcripts, and then that allows them to basically take over the plant cell. So, this modification, there's an arms race there too, right? And we do think that there's going to be some sort of sensing mechanism. That's why we're getting more interested in that cat methylation event. We're thinking that there's going to be some arms race both with M6A and that methylation of M7 guanosine. That's very, very preliminary, and I can't talk more about it other than just to say there's definitely an arms race. Pathogens are definitely using the methylation machinery against plants and animals as well. I mean, like everything I talk about in plants is true in animals, right? They have the exact same proteins. There's one-for-one orthologs, other than when it comes to the readers. And then what happened is in plant transcriptomes, I didn't say this, but I'll say it very, very quickly. Plant transcriptomes have 3 times as many readers as compared to the animal systems. Like Arabidopsis has 12, humans have 5, right? So, and then there's other plants that have up to 18 or 20. So usually there's about 3 times as many readers in plants, because they're probably sub-functionalizing readers to specific stress responses, and we see that in our data. So thank you. Oh, I'll take one more question.
This is a follow-up to what Andrew just asked. So in terms of biotic stress, you're seeing effects on bacteria, but also increased resistance to fungal pathogens. And I know that's somewhat surprising, right? Because often those are kind of play off each other.
That's correct.
Your JA or SA pathways. So does that tell you something about the news number of guys' devices?
No, we were surprised because they show all the plants show all the classical hallmarks of bacterial resistance, but then you treat them with. So we didn't think they were going to be more fungally resistant, and then they were. So we're extremely surprised, and we're like combing back through all of our data right now to like figure out how it is. And what it looks like is it's probably a couple of transcription factors are going the same way, right? To then upregulate both responses the proper way. And that's how they're more resistant. So, I mean, like I said, we need to do a lot more work on that overexpression of the eraser, because if it truly does make them more pathogen resistant, not just weirdo Arabidopsis, right, in a perfect condition, but like if we can move that into the field, then that's great. That's great.
If there are no more questions, thanks, Ryan.
This has been a production of Cornell University, on the web at cornell.edu.